let's talk about receptors. So the reason this is going to be important is when we look at the whole animal and how the animal functions in relation to metabolism. One thing that we've been talking about a bit is how the brain can really only run on glucose and ketone bodies. The entire purpose of creating ketone bodies in times of starvation is that it can be less costly than gluconeogenesis, but it's mainly because the brain needs glucose. The brain is like a picky child that only wants sugar. And so, because this is the case, essentially the body will spend all of its energy and will try to supply the brain with glucose until your dying breath. The brain is the last organ that's going to be supplied. As a result of this, sort of evolutionarily, the liver has developed in order to sacrifice itself altruistically to supply the rest of the body. So we talk about the liver undergoing gluconeogenesis and then exporting that glucose into the blood for the rest of the body. But how is that possible? Well, it's all to do with receptors. And you'll have to bear with me through this because we're going to have to talk a little bit about some biochemistry and some Michaelis-Menten kinetics in order to sort of understand how receptors play a really important role in this process. So when I say Michaelis-Menten kinetics, we're not going to go really into detail, but the important part is about binding. So when you think about enzymes or receptors or even molecules binding to each other, the most basic way that you can think about this is in a chemical reaction, right? So something like sodium chloride. Well, why do sodium and chloride bind each other? It's because one of them is positive and one of them is negative. This same principle can be applied to more complicated molecules like receptors, like enzymes. And the important part to remember is that these are all proteins. They have positive and negative domains. They have nonpolar or fatty domains. These are all of this is determined by the characteristic of the amino acid that makes up the proteins. Now, you're not going to know just by being given the name of a protein and enzyme, like glycogen phosphorylase, what the domains are like or what it binds. As far as the substrate, it gives you a pretty good idea. Glycogen phosphorylase, it probably phosphorylates glycogen, and it breaks it down to glucose. So in the same way that enzymes bind their substrates in order to undergo a chemical reaction, receptors also bind substrates, ligands of a sort, but their reaction is transporting them in the cell or causing some sort of intracellular signaling cascade rather than undergoing a strict chemical reaction. So if we think about the glucose in the bloodstream, let's consider this a blood vessel. The blood supply of the body is supplying lots of different cells. So you might have a muscle cell here, you might have a brain cell here, you might have liver. Okay, and we know from our general cell biology that these cells are made up of a lipid bilayer surrounding them. That means that they're selectively permeable, only certain things can get across. In this particular case, glucose is a little too large and a little too polar to get across the membrane, right? We know that fats can dissolve across the membrane because of those really thick fatty tails on the inside of the bilayer. But glucose has some hydroxyl groups. A hydroxyl is just an OH. And if we remember from chemistry that oxygen is extremely electronegative, that means that it likes to pull the electrons close around it. And that gives this particular group on glucose a slight charge. It's not quite ionic, but it makes it polar, which means that it will, for example, dissolve in water, just like your table sugar. So in order to get glucose from the blood vessel into any one of these cells, these tissues, it needs a protein on the surface of the cell in order to transport it across. This transporter is different in each of these tissues. It is the same sort of general protein. It's called the GLUC transporter, GLUC, and that is a protein that is on the cell surface. I believe it's a channel. That just means that you can think of it a bit like a tube, but that tube is the correct diameter and polarity to only allow glucose through it. So it's not like just poking a hole in the membrane. It's a little more selective than that. What makes brain, liver, and muscle different is that there are different isoforms of this GLUC transporter at the membranes of each of these. Okay, so in muscle cell, you might have something called GLUC1, and in liver, you might have something called GLUC3. It means that it's the same basic protein, but it has a slightly different domain that changes how well it binds. Okay, so we've got the GLUC transporter here for liver. Okay, we'll call this GLUC3. And then here in brain tissue, we'll call this GLUC2. Okay, so they're both going to transport glucose. They're both going to use the same mechanism to transport glucose, but the important bit is that their binding affinity is slightly different. Okay, so the reason that this is important is because we've been talking about, say, mass action in relation to how fast a reaction goes forwards or backwards, mass action being the concentration of substrate. So one way that we can look at this is by saying what happens if the glucose level in the blood is very, very, very low. Okay, let's say that your concentration of glucose in the blood, let's say that you're in a fasting state, and it's around 1 millimolar. Okay, the actual number isn't that important, but for our purposes, this is a very small concentration in the blood. Well, essentially, the way that this whole system is designed is that the brain is sensitive to 1 millimolar glucose concentration in the blood. And even at very, very low concentrations, I said the brain is like a picky, greedy child. The brain will continue to take up glucose at 1 millimolar concentration through this glucose transporter, and it will continue to burn it. The reason this is the case, if anyone's curious, is because the brain is undergoing all these action potentials, and it takes a lot of energy in order to depolarize and repolarize those cells constantly. So most of the energy in the body is used to create or maintain gradients, and because the brain is a large collection of neurons, it simply has to do that a lot more than the rest of the body. Alright, now what happens if we say have a meal, you're no longer in a state of starvation, and the glucose concentration increases to 10 millimolar. Okay. Well, what that means now is that the liver is sensitive to, say, a 10 millimolar concentration of glucose, and the liver will now start taking up glucose, because what this transporter allows is that the liver is only sensitive to glucose concentration in the blood in times when the body is well fed, which means that it will take up glucose and it will be able to store it for later. That's really the liver's function, is just storage. It's the rate of its Krebs cycle and its electron transport chain, its metabolism for the purposes of its own cell. The rate of those reactions doesn't really change in relation to whether the body is fed or fasting. Okay, what's really going to change is whether the glucose is being taken up and stored as glycogen or fatty acid, or whether the glycogen and fatty acid are being broken down so that glucose can be exported to the bloodstream. Okay, so if glucose concentration drops below a certain threshold level, it might be 5, it might be 6, the number is a bit arbitrary, then this transporter will no longer be sensitive to glucose concentration, and it simply, the binding efficacy, the affinity of this particular transporter, this isoform of the GLUC transporter, is too low, and unless you flood the system with glucose, it just won't even recognize that it's there. And that's really evolutionarily advantageous, because it lets the rest of the body take what glucose it needs, and then the liver only takes when there's excess, so that it can store it for later. If anyone's curious, the muscle also has glucose transporter, and it's sort of an intermediate, we'll call this GLUC1. It's sort of an intermediate binding affinity, so it might absorb glucose at 4 millimolar, or 3, something like that. Okay. So I hope that clears up a little bit. The next thing that I'm going to talk about in the subsequent video is binding efficacy in relation to enzymes, because it's very closely linked to this concept of receptors.